Welcome back to the deep dive. Today we're unpacking a piece of research that, honestly feels like it was ripped straight out of a sci-fi novel. But it is very real, very new science.
It is. We are looking at a study from Cell. This is, 2026, so it's basically hot off the press by Gal and colleagues. And the topic is, well, it's a heavy one, glioblastoma.
Right, GBM. For anyone who doesn't know, it's the most lethal primary cancer of the central nervous system.
And it's so terrifying, not just because of how fast it grows, but because it's a master of disguise. It creates what we call an immunosuppressive environment.
It basically shuts down the body's own police force, the immune system.
Exactly. So it can just grow completely unchecked. It built its own fortress. Yeah. And when we try to attack that fortress, we're usually thinking from the inside, right?
Yeah. Surgery on the brain, radiation on the brain, drugs that have to somehow get into the brain.
But this paper, it flips that completely on its head. It asks a question that, frankly, sounds a little nuts.
It does. It asks, what if the key to fighting a tumor inside the brain is actually blocking blood vessels outside the brain?
That's the hook. The title is Meningeal Blood Vessel Blockage Enhances Anti-Glioblastoma Immunity. And our mission today is to just, walk through that sentence by sentence, mechanism by mechanism.
Okay, so let's get into this hack. But before we do, I need an anatomy lesson because my alarm bells are ringing.
I'm sure they are.
When I hear blood vessel blockage and brain in the same sentence, I think stroke, I think ischemia. Aren't we just killing brain tissue?
That is the first and most critical question. And to get it, you have to picture what the paper calls the dual plumbing system of the head.
Dual plumbing.
Imagine two totally separate sets of pipes. First, you have the internal carotid artery, the ICA. This is the main line. It runs deep, and it feeds the brain parenchyma.
Which is the actual brain tissue, neurons, thinking, all that stuff.
And where the tumor lives, unfortunately. The ICA is the pipeline for the brain and the tumor. This paper does not touch the ICA.
Okay, so the brain's food supply and the tumor's is safe. That's a relief.
Completely safe. Then you have the second system, the external carotid artery, the ECA. This one's more superficial. It feeds your face, your scalp, and this is the crucial part, the dura mater.
The dura. That's the thick, leathery sac that covers the brain.
Exactly. Think of the brain as the living room of a house, and the dura is the roof and siding. The ICA feeds the living room. The ECA feeds the siding.
So this procedure, meningeal blood vessel blockage, or MBB, it's about tying off the ECA.
That's it. They ligate it, tie it off, and they did a ton of safety checks. MRIs, Nissl staining to look at the neurons.
What did they find?
Zero ischemia. No stroke damage at all. The neurons were perfectly healthy because, again, their supply line, the ICA, was completely untouched.
Okay, so it's safe. We've cut blood flow to the covering, but not the brain. But here's the mystery. The tumor is inside, eating from the ICA. Cutting the ECA doesn't starve the tumor.
You've hit the nail on the head. That is the central question of the entire paper.
So if we're not starving it, why on earth does the tumor stop growing?
And that is the aha moment. It's not about starving the tumor of nutrients. It's about changing the entire immune neighborhood. To get this, we have to meet the cast of characters living in the dura.
The microscopic tenants.
Right, because the dura isn't just a wrapper, it's a bustling immune hub. The researchers did single-cell RNA sequencing, basically a census of every cell. And it is teeming with macrophages. They call them border-associated macrophages. BAMs. But, and this is the core discovery, there are two different types, two distinct clans. It's like a turf war.
Okay, so who are the families in this war?
First, you have the CBAMs, the C's are for circulation derived. These guys come from the bloodstream. They're basically monocytes that hop off the blood vessels, the ECA vessels, and into the dura.
Okay, so the CBAMs are like tourists. They take the highway, the ECA, into town. They hide out for a bit, but they're always being replaced by new arrivals.
That is a perfect analogy. They are transient, short-lived. They're just passing through. So they're not great at, teaching the immune system any deep lessons about the local area.
Got it. And the other clan?
Those are the RBAMs. Ours is for resident. These are the locals. They live in the dura full-time. They have different markers. And crucially, they have really high levels of a receptor called FcRn.
We'll get to that receptor, I'm sure. But for now, the RBAMs are the local guardians. They know the neighborhood.
Exactly. And because they're residents, they are way better at presenting antigens, at teaching the immune system what to attack.
So in a normal brain, who's winning this turf war? The tourists or the locals?
The tourists. The CBAMs are constantly flooding in from the blood, crowding out the RBAMs. The highway is wide open.
And here's the twist. when they do the MBB surgery and cut the ECA.
They close the highway. The tourists can't get into town anymore.
Right, so the CBAM population just crashes.
Yeah.
But the RBAMs, they're already there. They don't need the highway.
And all of a sudden, the whole balance of power shifts. The tourists are gone, and the local guardians take over.
Okay, this feels like a perfect spot to pause. We've covered a lot of ground pretty fast. Let's just do a quick recap to make sure we've got the board set.
Let's do it.
All right, recap number 1. First, the anatomy. We've got 2 plumbing systems. ICA feeds the brain, ECA feeds the dura, the covering. Second, the surgery. MB ties off the ECA. It's safe. It doesn't starve the brain.
Right.
Third, the mystery. It doesn't starve the tumor either. Instead, it changes the immune environment in the dura.
The neighborhood changes.
Fourth, the players. We have the CBAMs, the tourists from the blood, and the RBAMs, the resident locals.
The 2 families.
And finally, the effect. When you cut the ECA, you stop the tourists from showing up and the locals take over.
Perfect summary. But now we have to answer why. Why does getting rid of the tourists suddenly make the locals boom?
Right. I mean, usually cutting off blood kills things, doesn't make them grow. How does starving the dura help the good guys, the RBAMs?
It all comes down to resource competition. Think of it like the Hunger Games of the dura. Both of these macrophage families, tourists and locals, are fighting over one specific resource, a protein called CSF1.
Colony stimulating factor 1. It's basically macrophage food.
It's prima macrophage food. It's what they need to survive and to multiply.
Right.
And it's made by cells in the dura called fibroblasts. The key thing is the supply of this food is constant. It's like a buffet that's always full.
I think I see where this is going. Before the surgery, you have all those CBAM tourists flooding in.
Constantly.
And they're just mobbing the buffet, eating all the CSF1.
They are gluttons. They eat almost all of it, leaving just scraps for the RBAMs. The locals are basically starving, so they can't multiply.
Then we do the surgery. ME, we shut the highway. The tourists stop coming.
And suddenly the buffet is overflowing. The study found that CSF1 levels in the dura doubled just 3 days after the surgery. With no one to eat it, the food just piles up.
And the RBAMs, who are still there, finally have an all-you-can-eat pass.
They feast and they proliferate. They start dividing like crazy. The population of these local guardians just explodes.
It's a supply and demand hack. You're not adding food, you're just getting rid of the competition. Did they prove this was the actual mechanism?
They did, and it was beautiful. They did a diphtheria toxin experiment. They used a genetic trick to kill only the CBAMs without surgery.
So the highway was still open, but the tourists were just gone.
Exactly. And guess what happened? The RBAMs expanded, just like with the surgery. It proved it was the presence of the CBAMs holding them back, not the blood flow itself.
That's so clever.
And then to lock it down, they did the surgery, but then they added an antibody that blocks CSF1. So they closed the highway and locked the buffet.
And then, no expansion.
No expansion. It proved the whole chain of events. Cut the vessel, get rid of the CBAMs, raise the CSF1, which then feeds the RBAMs.
Okay, so the hack works. We now have a massive army of RBAMs, the local guardians, controlling the dura. But we still have a problem. The tumor is inside the brain. The army is outside. How do they fight an enemy across the border?
This is the next phase. This is the weapon. The paper calls it the dura-CSF-TME axis. It sounds complicated, but it's just a kill chain, a series of steps.
Okay, let's walk through the chain. Step 1.
Step 1, antigen capture. The brain is floating in cerebrospinal fluid, CSF. This fluid is constantly circulating, and it acts like a sewer system. It washes away waste.
Including tumor trash.
Exactly. As the tumor grows and cells die, bits of it, antigens, get flushed out into the CSF, and that fluid washes right over the dura.
So the dura is like a filter, catching the evidence.
It is. And this is where the RBAMs shine. Remember I said they have high levels of that FcRn receptor?
I do. The neonatal Fc receptor.
So FcRn acts like a specialized pair of handcuffs. It's incredibly good at grabbing onto immune complexes, which are just tumor antigens that have already been tagged by antibodies. The RBAMs use FcRn to snatch this evidence out of the fluid.
It's like an intelligence officer sifting through the trash, finding the wanted poster for the tumor.
That's a perfect image. And once it finds that poster, it gets triggered. The paper also found that tumor DNA, double-stranded DNA, gets into the CSF. When the RBAM eats this DNA, it triggers an internal alarm system, the CGA sting pathway.
Sting. Sounds aggressive.
It is. It stands for stimulator of interferon genes. It's a red alert. It wakes the cell up and tells it we are under attack. So now you have an RBAM that's angry, awake, and holding the enemy's wanted poster.
But the macrophage isn't the killer, it's the intel officer. Who does it give the poster to?
To the assassins. The dura is also patrolled by CD8 plus P cells. These are the snipers of the immune system. The RBAM holds up the antigen and shows it to a T cell. It's a handoff. This is the guy. Go get him.
And the T cells can cross the border.
That is the crucial leap. These activated, trained T cells migrate from the dura across the barrier and into the brain. They swarm the tumor microenvironment, or TME.
And when they get there, they're ready to fight.
Oh, yeah. Data show the T cells inside the tumors of the MBD mice were loaded for bear. They were full of granzyme B and interferon gamma, the chemical weapons they use to kill cancer cells.
And they weren't tired. Sometimes T cells get exhausted.
They were less exhausted. That's a huge deal in cancer immunotherapy. They just kept fighting.
This is incredible. So you're basically using the dura as a secret military training camp.
That's exactly what it is.
You cut the supply lines to clear out the riffraff. You feed your elite trainers, the RBAMs. They grab intel from the wastewater, train the special forces, the T cells, and then send them across the border to win the war.
You've just synthesized the entire mechanism, from the outside plumbing to the war on the inside.
Okay, let's lock that in with our recap number 2.
Go sure.
The mechanism of action. First, the food war. MBE cuts the ECA, stops the tourist CBAMs, which leads to a surplus of CSF1 food.
Sure.
Second, the expansion. The local RBAMs feast on that CSF1 and multiply like crazy.
A local army.
Third, the intel. The RBAMs use their special FcRn receptors to grab tumor trash from the CSF.
The wanted posters.
Fourth, the alarm. They get activated by the S-sting pathway. And fifth, the handoff. They show the evidence to the CD8 plus T cells.
Training the assassins.
And finally, the attack. Those T cells migrate into the brain and destroy the tumor.
That's the whole cascade. Beautifully put.
But the big question, does it actually work? I mean, understanding the pathway is one thing, but did they live longer?
They did, significantly. In one model, the control mice with GBM survived for about 27 days. The mice that got the MBB surgery, they survived for about 38 days.
That's almost a 40% increase in a disease like glioblastoma, that is. That's massive.
It's substantial, and that's before they even tried combining it with other things. But first, they did this really important sanity check.
The nude mouse check.
Yes. They did the exact same surgery on nude mice, which are genetically engineered to have no T cells, no adaptive immune system.
So if the surgery just worked by starving something, it should still work. But if it works by training T cells...
It should fail. And it did. The surgery did absolutely nothing in the nude mice. The tumors grow just as fast. It's the smoking gun that proves the entire effect is driven by the immune system.
That is fantastic validation. So what about those combination therapies?
They paired MBB with an anti-PD-1 drug, which is a common immunotherapy that takes the brakes off T cells. The effect was synergistic. They saw some mice completely cured.
Cured, like long-term survivors.
Long-term survivors, no detectable tumor. They also tried just injecting extra CSF1 into the dura along with the surgery to really supercharge the RBAMs. That worked incredibly well, too.
This is all in mice, of course. The classic disclaimer, is there any evidence this biology exists in humans?
There is, and it's really compelling. They got dura samples from human GBM patients and did the same sequencing, and they found the exact same two populations.
The tourists and the locals, CBAMs and RBAMs.
Both there. And here's the kicker. They found that human patients who naturally had more RBAMs in their dura lived significantly longer.
Wow. So the mechanism holds. Having more local guardians is protective.
It strongly suggests that if we can find a way to boost them, like with MBB, we'd see a benefit in people.
And is this surgery even realistic for people?
It's surprisingly realistic. There's a procedure neurosurgeons already do called MNA embolization, middle meningeal artery embolization.
What for?
To treat chronic subdural hematomas, basically bleeding in the dura. They go in with a catheter, find the artery feeding the dura, and block it. It's minimally invasive.
So we don't need to invent new tech. We just need to repurpose an existing plumbing procedure for a totally new reason. Immunotherapy.
Exactly. And that's why this paper is making such huge waves. It connects basic science directly to a practical existing clinical procedure.
So when we zoom out, this is a real paradigm shift. We used to think of the brain as immune privileged, a fortress with a drawbridge, a
Right, that the immune system wasn't allowed in. This paper says, no, the brain has an embassy. The dura is that embassy. It's an immune interface where the brain and the body talk.
And the problem is that embassy is usually either too quiet or it's overrun by tourists who don't care.
Right. And MBB is the hack. You're manipulating the local environment to amplify the right voices. The authors actually call the dura an immunological amplifier.
Yeah.
MBB is just turning the volume knob up to 11.
Hacking the amplifier. I love that. It really connects all the dots, the blood vessels, the macrophages, the CSF, the T cells.
It's engineering the body to do the work for you instead of just trying to poison the cancer cell directly.
I want to leave our listeners with one last thought to chew on. We've talked about this for cancer, but the brain has other problems that involve trash removal.
That's where my mind goes too. Think about Alzheimer's disease. It's fundamentally a problem of waste accumulation amyloid plaques that the brain can't clear. And what are the brain's trash collectors? Macrophages.
So, if you can manipulate these dura macrophages, RBAMs, to be better at their jobs.
Could we treat neurodegeneration by tweaking the plumbing of the meninges? If cutting a vessel wakes up the immune system to fight cancer, could a similar trick help it clear the plaques of Alzheimer's? We may have just stumbled upon a control panel for brain health we never knew existed.
That is a wild and hopeful thought. Meningeal engineering.
It's a brand new frontier for neuroimmunology.
We want to hear from you. Check the show notes for a diagram of the RBAM eating the tumor antigen. You have to see it. And let us know. If you were a neurosurgeon, would you risk cutting a healthy artery to wake up the immune system? Is the hack worth it?
It's a huge question, but the data here is incredibly compelling.
Stay curious and keep diving deep.
Until next time.